this video, we're going to look at question number 14 from GATE Biotechnology 2023 paper. The question is, determine the correctness or otherwise of the following assertion A and the reason R. So the assertion is chromosome mutations can change the structure of chromosomes, and the reason R is all chromosome mutations arise due to non-disjunction of chromosomes during mitosis or meiosis. So the given options are both A and R are false. Option B, A is true but R is false. Option C, both A and R are true and R is the correct reason for A. And option D, both A and R are true but R is not the correct reason for A. This represents a karyogram of the diploid human genome with 23 pairs of chromosomes that includes 22 pairs of autosomes and 2 sex chromosomes called allosomes. So chromosomes can be categorized based on their morphology as metacentric, where the centromere is in the middle and there are equally sized P and Q arms on both sides. Submetacentric is one arm, which is the P arm, is slightly shorter than the other longer arm, which is the Q arm. Acrocentric is where one arm is very short and the other is long. And telocentric is where the centromere is towards the end of the chromosome, where the short arm, which is the P arm, is not visible or it's just barely visible. Let's now get into chromosomal mutations. So these chromosomal mutations or chromosomal aberrations include rearrangements, aneuploids, and polyploids. So chromosome rearrangements alter chromosome structure. So rearrangements could include deletions, insertions, duplication events, which we'll be looking in detail in the next few slides. And aneuploidy is an increase or decrease in the number of individual chromosomes. And polyploidy is the presence of more than 2 sets of chromosomes. So chromosome variation overall plays an important role in evolution and also contributes to a lot of diseases. Here's a visual representation of chromosomal mutations that consists of chromosomal rearrangements, aneuploids, and polyploids. So duplications, trisomy, and autotriploids are just examples of each category of the mutation, which we'll look further into detail one at a time in the next upcoming slides. The first kind of abnormality are chromosomal rearrangements. So they do not increase or change the number of copies of the chromosome, but just alters the structure. For example, in duplication, some segments like E and F get duplicated and would have more copies of a gene in this region. So in terms of recombination, non-disjunction is the failure of homologs of chromosomes or sister chromatids to separate properly during cell division, that is in mitosis or meiosis. So during prophase 1 of meiosis, in an individual heterozygous for a duplication, that is just one of the pair of the chromosome has duplicates while the other is normal. The chromosome with the duplicated segment forms a loop-like structure when it pairs with its homologous chromosome. So this looping out is just a visual representation of the homologous recombination process where genetic materials exchange between two homologous chromosomes. So the phenomenon of looping out is one of the ways in which genetic diversity is generated during meiosis. So it allows for the exchange of genetic material between homologous chromosomes, leading to the creation of genetically unique gametes. So the details of how the loop formation occurs would involve molecular processes such as the formation and resolution of junctions during homologous recombination. So it's just that the two same segments of the chromosome, they pair with each other. So since this normal chromosome doesn't have these duplication events, these are just extra segments of the chromosome and it just gets looped out. So it leads to the formation of abnormal gametes. So let's look at an example in Drosophila where chromosomal duplication events has led to phenotypical abnormalities. So one phenotype that has been analyzed in Drosophila with respect to duplications is bar eye. So the eye of the fly is normally an elongated oval shape, whereas the bar eye phenotype is much thinner. So when the chromosomes of males with bar eye are analyzed, a duplication in the region 16A of the chromosome is detected. So wild type fruit flies have normal sized eyes. So flies that are heterozygous, that is just one copy of the chromosome of the duplication event, has like a smaller eye. So it's the same as when you could see a duplication even in both the copies of the chromosome. They have a smaller eye. So flies with double bar have 3 copies of the duplication and much smaller bar-shaped eyes. So this just shows how these rearrangement in the chromosomes lead to visible phenotypical abnormalities. Unbalanced gene dosages, that is extra copies of the gene, also leads to developmental abnormalities. And then rearrangements also include deletion of chromosomal segments. And again, this is how homologous pairs of chromosomes with heterozygous deletion recombines, again forming abnormal daughter chromosomes. So in an individual heterozygous for deletion, the normal chromosome loops out during chromosome pairing in prophase 1 of meiosis. Another form of rearrangement is inversion. So inversion could be either paracentric, which involves inversion within one of the arms of the chromosome, as shown here in this figure. So the inversion just happens within the segments, that is in the longer arm of the chromosome, and it doesn't involve the segments that are on the other side of the centromere. So the other form of inversion is pericentric inversion, where segments across both the sides of the centromere are involved. So these events also lead to non-disjunction during meiosis. An example of inversion in the context of evolution is where chromosome 4 differs in humans and chimpanzees in a pericentric inversion. So we could see that the chromosomal segments here, which are on either side of the centromere, they seem to be inverted in humans and chimpanzees. Like if you could see here, the red band over here is on the shorter arm of the chromosome, uh, and whereas in the chimpanzees this red band is in the longer arm of the chromosome. So it has just been inverted in both across both these species. So this is an inversion event that you observe across species. The next type of rearrangement is translocation. So reciprocal translocation is when part of one chromosome is exchanged with a different chromosome. So you could see 2 different chromosomes here, and you can see that a part of this, the QRG region, like the QR segment from this chromosome, has been exchanged with the EF segment of this chromosome. So you could see that these 2 chromosomes have been translocated. So Robertsonian translocation is where the short arm of one acrocentric chromosome is exchanged with the long arm of another acrocentric chromosome. So this type of translocation is usually seen in chromosomes 13, 14, 15, 21 and 22 in humans because the ends of the short arm have similar repetitive DNA. So in an individual heterozygous for a reciprocal translocation, cross-like structures form in a homologous pairing, which also leads to like the formation of abnormal chromosomes in the gametes. So human chromosome 2 contains a Robertsonian translocation that is not present in chimpanzees, gorillas, or in orangutans. So G banding, that's chromosomal banding, reveals that a Robertsonian translocation in a human ancestor switched the long and short arms of the true acrocentric chromosomes that are still found in other 3 primates. So this translocation created the large metacentric human chromosome. So the chromosome 2. So the human chromosome 2 has 2 almost equally sized chromosomal arms. So hence it's metacentric. Whereas in other species, you can find one small, like it's an acrocentric chromosome. Another type of chromosomal abnormality is aneuploidy, which is the increase or the decrease in the number of individual chromosomes. So it occurs due to several reasons like deletion of the centromere during mitosis and meiosis, or translocation, and mechanisms that lead to non-disjunction during these cell division like meiosis and mitosis. So there are different types of aneuploidy. So one is nullisomy, which is the loss of both members of a homologous pair of chromosomes. So let's say if the total number of chromosomes in a diploid genome is denoted as 2n, so nullisomy is 2n minus 2, where like 2 pairs of a single chromosome has been deleted. For example, in humans, let's say we have 23 pairs of chromosomes, which is 2 into 23, And so let's say we have two copies of chromosome 1. So both of these copies have been deleted, and we have all the other chromosomes except for chromosome 1. So that is when you call this condition nullisomy. And then monosomy is the loss of just a single chromosome. Like in the previous example that we saw that both the copies of chromosome 1 had been deleted. But in case of monosomy, just one of the copies of chromosome 1 is deleted. So it's denoted as 2n minus 1. And then we have trisomy, which is the gain of a single chromosome. So let's say we have an extra copy of chromosome 1. So in total, we have 3 copies of chromosome 1 instead of 2. So it's denoted by 2n plus 1. And then in case of tetrasomy, which is the gain of 2 homologous chromosomes. So hence it's denoted as 2n plus 2. So like we have an extra pair of copy. So in this case, we have 2 more extra chromosome 1. So in total, we have 4 chromosome 1. So these are the types of aneuploidy. So like we saw earlier, aneuploids can be produced through non-disjunction in meiosis and mitosis. So the gametes that result from meiosis with a parent with abnormal chromosomes with non-disjunction combine with a normal gamete with a blue chromosome from another parent. So this results from normal meiosis to produce the zygotes. So for example here, non-disjunction happens and we have two products of meiosis 1, and this segregates like these. So we have 2 copies and we have 2 copies of one chromosome instead of having just one of the red copies of the chromosome. And the other case is where no copies are found from this cell. So when this one fertilizes with a normal gamete, so the zygotes would be trisomic, that is 2n plus 1. So we would have 2 copies from parent, the red parent, and we would have one copy from the blue parent. So in case the normal gamete fertilizes with this particular gamete from the red parent, monosomic zygotes are formed. That is, no chromosomes from the red parent and just one copy of the chromosome from the normal parent. So this is an overview of how aneuploidy is formed. So in the earlier example, we saw non-disjunction in meiosis 1, so which led to 2 copies of this in one of the cells and no copy in the other cell. So in this example, the non-disjunction happens during meiosis 2, while meiosis 1 forms normal cells. So in this case, we'd have one daughter cell with both the copies of from the parent red and the other one with no copies. So when these fertilize with a normal gamete, we find trisomic and monosomic zygotes. So whereas the other one with one copy from each of one copy of the red chromosome from the red parent in each cell fertilizes with the normal gamete, it leads to the formation of normal diploid zygotes. And now just to brush up the concept, so let's say a diploid organism has 2n is equal to 48 chromosomes. So how many chromosomes will be found in a trisomic member of this species? So we learned that trisomy is a condition where the total number of chromosomes is represented by 2n plus 1. So in this case 2n is 48. So 2n plus 1 would be 49. So the trisomic individual would be having 49 chromosomes. So here's an example of the effects of aneuploidy in plants. So mutant capsules in Jimson weed, that is Datura stramonium, result from different trisomies. So each type of capsule is a phenotype that is trisomic for a different chromosome. So the structure of the seed case in the wild type with 2n chromosomes would look like this, whereas the trisomic seeds which has like trisomy in like different chromosomes look like any of these phenotypes. So they're either rolled or glossy or elongated or like a globe, and like all many different phenotypes. So aneuploidy in humans can lead to several abnormalities. For example, in case of sex chromosome aneuploids, one example of a condition is Turner syndrome, where the female is born with only one copy of the X chromosome. So this is the loss of a single chromosome, and so it is monosomy, which is 2n minus 1. So in case of Klinefelter syndrome, so this is where a male is born with an extra X chromosome. So it is 2n plus 1, and this is an example of a trisomy. So aneuploidy in the autosomes can also lead to diseases. For example, trisomy in the chromosome 21 of humans leads to a condition called Down syndrome. So primary Down syndrome is where 75% random non-disjunction happens during the egg formation, whereas familial Down syndrome is where Robertsonian translocation happens between chromosomes 14 and 21. So, primary Down syndrome is caused by the presence of 3 copies of chromosome 1. So, this is a karyotype of a person who has primary Down syndrome, and you can see that all the other chromosomes are normal, whereas you can see 3 copies of chromosome 21. So, whereas in case of familial Down syndrome, the translocation of chromosome 21 onto another chromosome has occurred here. So, here you can only see the short arm of chromosome 21. So, the long arm of chromosome 21 is attached to chromosome 15. So, this karyotype is from a translocation carrier who is phenotypically normal but is at increased risk for producing children with Down syndrome. Other examples of autosomal aneuploids are trisomy of chromosome 18 that causes Edwards syndrome, and trisomy in chromosome 13 which causes Patau syndrome, and trisomy in chromosome 8. It is also usually seen that there is a correlation between maternal age and giving birth to a child with autosomal aneuploidy. So this plot here is an example of older the maternal age, higher the cases of Down syndrome. So there is a correlation between maternal age and the incidence of giving birth to such aneuploid children. Uniparental disomy is another term in aneuploidy. So this is an abnormality where both chromosomes are inherited from the same parent. So so that would be represented as n plus 1. So the haploid genome would, like, it would just have n plus 1, which comes from the same parent, as denoted as two red chromosomes over here. So this is an example of uniparental disomy. So there's no chromosome from the other parent. So we have so far seen about chromosomal rearrangements and aneuploidy. So another form of chromosomal abnormality is polyploidy. So polyploidy is the presence of more than 2 sets of chromosomes. So it could be of 2 types, autopolyploidy or allopolyploidy. So autopolyploidy can arise through non-disjunction in mitosis and meiosis, and this occurs when organisms have more than 2 sets of chromosomes from the same species. So that is, like, in general, like for example in humans we have 2n chromosomes. So polyploidy would result in 3n or 4n chromosomes. So autopolyploids can originate from issues that arise during mitosis, such as doubling of the chromosomes without any cell division. So this would result in a tetraploid organism, that is 4n, from a diploid organism. So the other one is allopolyploidy. So this occurs when an organism has more than 2 sets of chromosomes from different species. So an example of allopolyploidy is Gossypium, a species of cotton, which are hybrids formed from 2 diploid species of cotton. So, in conclusion, chromosomal variations play an important role in evolution, giving rise to new functionalities and even new species. Now, coming back to our question, so let's look at the assertion. So, chromosome mutations can change the structure of chromosome. So, this statement is true because we saw that events such as deletions, duplications, and translocations, they all change the structure of the chromosome. So, the assertion A is correct. And the given reason, the statement is all chromosome mutations arise due to non-disjunction of chromosomes during mitosis and meiosis. So here this statement in itself is not true. It is false because non-disjunction is one of the reasons that lead to such chromosomal abnormalities like ploidy and autoploidy. So, whereas there are several other reasons that can also contribute to chromosomal mutations, such as environmental factors like mutagens and fault in the DNA repair mechanism, and point mutation and so on. So the reason the statement itself is false. So the correct answer to this question would be option B, where A is true but R is false.